My name is Brennan Weber, and for microteaching, I'm doing a lesson plan over mitosis. So mitosis is a type of cell division that results in two daughter cells, each having the same number and kind of chromosomes as the parent nucleus, typical of ordinary tissue growth. So mitosis, even though they sound the same, is significantly different than meiosis, and if not corrected in time, mistakes made during mitosis can result in changes in the DNA. But before we begin, there's going to be a few things we need to go over. First is the difference between prokaryotes and eukaryotes. Prokaryotes have no nucleus or any other membrane organelle. Mitosis does not occur in prokaryotes since they do not have a nucleus, but they do divide in different ways. Prokaryotes include single-celled bacteria and cyanobacteria. Eukaryotes, on the other hand, can be single or multicellular, and they have membrane-bound organelles such as the nucleus, the mitochondria, and endoplasmic reticulum. In eukaryotes, genetic material is DNA in the form of chromosomes, which are contained within a distinct nucleus. Also, we need to talk about the role of the nucleus. It has two major functions. It controls hereditary materials and also coordinates cell activities. And something that's kind of important with DNA is the nucleus makes ribosomes, and ribosomes play a big part in DNA structure. So, getting into mitosis, it has four phases: prophase, metaphase, anaphase, and telophase. An easy way to remember that is just that it spells out PMAT. I'll go in that order. So prophase is the first stage of cell division before metaphase, during which the chromosomes become visible as paired chromatids and the nuclear envelope disappears. At this point, chromosomes become coiled and can be viewed under a light microscope. The second stage is metaphase. The chromosomes become attached to the spindle fibers, and at this point they're most highly coiled and combined, which makes them the easiest to see. The third stage is anaphase. The chromosomes move away from one another to opposite poles of the spindle. So you kind of think of that as like a middle school dance. Everyone just boys gravitate to one side of the wall, girls gravitate to the other side. It's just them separating. And the final phase is telophase. The chromatids or chromosomes move to opposite ends of the cell and two nuclei are formed. Once the two nuclei are formed, the cell essentially gets pinched and splits into two different cells. So, just to sum everything up, mitosis is a type of cellular division that consists of four phases: prophase, metaphase, anaphase, and telophase, and each step provides unique instruction when it comes to making a new cell. I have a video that we can watch. It is 8 minutes long.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it, and a few days later, it's gone. It's all healed, and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails, and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror, and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes, very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PNAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PNAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there. And it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are they're moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Alright, so, hope that video was helpful. Back to PowerPoint.
Well, Bill,
I don't know.
Arlene says I gotta sleep in the barn tonight. Says I shouldn't have bumped off.
So now we're going to do our group activity. I'll let you guys pick your own groups.
My partner.
So during this activity, I'm just going to have you guys on a piece of paper. You can use this piece of paper or your own. Write down the 4 phases of the 4 steps to mitosis and briefly describe what they do.
I took notes.
Here's some help if you need it.
Oh, and we have a PowerPoint work on. So the 4 steps. So PMAT. So prophase, I have the chromosomes. Chromatids and are visible? That doesn't, yeah, that sounds right. Okay. So, oh, they become coiled. I forgot to write coiled. They can be viewed under a microscope, though, so that's the visible part. The next is metaphase. So I have the chromosomes combined with the spindle fibers up the middle of the cell, or in the middle of the cell. Anaphase, which is when the chromosomes separate thanks to the spindle. They go to opposite ends. They separate to opposite ends thanks to the spindle. Last, telophase. So this is when they separate, they get pinched and split. they form their own nuclei. Look at us, guys.
Perfect. Even though you guys had the answer key, we're going to go ahead and just go over these answers on the board. So step 1 is prophase. Please excuse my terrible handwriting. Second stage is metaphase. Third is anaphase. And last is telophase. And like the video said, for prophase, the first step before metaphase, during which the chromosomes become visible and are paired to chromatids. Metaphase, they become attached to the spindle fibers in the middle of the cell. And anaphase is when they all separate and move away from each other. And then in telophase, they form 2 nuclei and separate. Now normally in a regular classroom I would have the students do an independent activity where they would essentially do the same thing but by themselves without the help of their notes or their peers, but for time's sake we're just going to skip that. The next thing that we have is going to be our exit slip, which there are 4 questions that you guys can just briefly talk about. Maybe use a few minutes to do that, and then we're going to do a KWL chart. So write one thing you know, one thing you learned, one thing you already knew, and one thing you want to learn. So you guys can go ahead and talk about that for a few minutes, or just write it.
I did not already know any of this. I did not already know any of this. But what did I learn? Well, I knew, I had heard of all of these phrases. I had no clue what order they went in or what they were particularly for. And there's a lot I wouldn't know.
See, I knew all this.
And it's okay if there's still things you don't want to know.
All of it.
All of it. All right.
What I want to learn, what is meiosis? Do they sound the same because they're close? I don't know. That could be something else because of...
Meiosis has to do with sex cells, so sperm and egg cells in the formation,
Sounds fun.
which would theoretically be the next lesson,
So it is kind of the same. Cell formation.
essentially.
Okay, cool.
Alright, like I said, normally we would do an independent activity and we would finish up with student-led discussion, such as questions and different topics that we could talk about if they're still confused. But like I said, for time's sake, and since we only have a few students in the audience, we're just going to cut it there. Thank you.
I think it's kind of